Hello everyone, my name is Matthew Leal, and I did my Keystone presentation through the College of Engineering, Biomedical Engineering Department, along with my partners Grace, Evgenia, Chancellor, and Ananya, under the direction of our advisor, Dr. David Boas. And the title of our presentation is Determining Brain Activation-Induced Blood Flow through Speckleplethysmography, also called SPG. And as a quick disclaimer, that's a very wordy title. And going into this project, most of us did not know anything about SPG. So we did a lot of background research prior to starting. And so because of that, I'm going to take a lot of the first part of the presentation through some of that background, and then later I'll get into more of what we actually did. So to start off, the motivation behind our work is neurodegenerative diseases such as Alzheimer's or dementia, and neurovascular diseases such as a stroke. Combined, these two types of diseases affect about 50 million Americans each year. We also know that they're all associated with some sort of change in blood flow, specifically usually a drop in brain activation-induced blood flow, which I'll talk more about in a minute. Now, normally we diagnose something like this with some medical imaging technique, a CT scan, an MRI scan. These aren't something that can be done continuously, and you need to go to a hospital to get this done. However, there's other ways that we look at blood flow throughout the body, usually through spectroscopy. For example, something that many of you may be familiar with is photoplethysmography, or PPG. It's found in a pulse oximeter. In your Apple Watch, it's the little green light on the bottom. And these measure volumetric changes in blood flow, and it does this by using an LED, which it shines through the body. That LED is then collected by a photodiode on the other side. And this photodiode measures the intensity of the light, which can show changes in how the light was absorbed by blood, which can show blood flow. And this works on your finger, your wrist, your ear, your forearm, but it's not known to work great in the body, in the brain. Specifically, it has a low signal-to-noise ratio. However, there's a relatively newer method called speckleplethysmography, or SPG, which measures basically the same thing, changes in blood flow. However, it uses a laser that's shown into the brain, and it collects a laser speckle pattern, which an example of one can be seen in red on the bottom. When you look at this speckle pattern, and you look at multiple of them, you can eventually begin to get an image, like the one on the right, that shows blood vessels. And you can look at this to get a measure called blood flow index, or BFI, which shows changes in blood flow in the brain. And in the brain, it has a very high signal-to-noise ratio, unlike PPG. So when you look at something like BFI, this is the raw signal that you get on the graph on the left. There's two things you're looking at here. There's the cardiac-induced blood flow, basically your heartbeat. That's the up and down on the graph that you see every second or so. But there's also brain activation-induced blood flow. And what I mean by this is more the graph on the right, which shows a typical hemodynamic response. So as you're looking at the response, there's an initial stimulus, and the brain responds by increasing blood flow to the brain, and then eventually it goes back down to normal. Now, if we were to look at that same graph on the left, it would look something like this. So there is very little change in the brain activation-induced blood flow in the raw data because of how much smaller it is than the amplitude of the raw data. So if there was a change or shift, you would never be able to see it. So for our project, we wanted to remove this cardiac component to just look at those small shifts in brain activation-induced blood flow. So to do this, we collected data of SPG from the brain and PPG from the finger and converted that to our raw data. The first problem we wanted to solve is we needed to get the beginning of each heartbeat to be able to add up that cardiac signal and subtract it. So there's a lot of different places this could have been taken from: the top peak, the second peak of those little bumps, the bottom of it, or the trough. We could have taken the first derivative and found the max of that. We eventually found the trough of each one to be the best at most accurately, from beat to beat, finding the exact start of each heartbeat. And once we had the start, we could find the mean cardiac output, which you can see on the left. And when you reinterpolate that back into the original data and subtract that, you get something on the right, which is 95% reduced from the original data. It looks a little messy, as you can see, kind of just a bunch of little bumps. So we thought we could do better than 95%. So we looked at all of the individual heartbeats, which you can see on the left. That is all 52 of them in this data set put together. And we decided to break them down into their individual components. This is called single value decomposition. And basically, we wanted to specifically filter out the ones related to cardiac blood flow. So when we did this, this was the new result we got. On the graph on the left, you can see the raw data in blue and the subtracted cardiac in orange, which would just show brain activation-induced blood flow. So we wanted to check that this actually worked and we could see a change in brain activation. So we convolved, on the graph on the right, the raw data with a sine wave, which acted like a hemodynamic response, so over the course of 5 to 20 seconds. And from the top, you can see there's very little change. But on the bottom, you can see that it did follow that sine wave. Here, to make it more clear, in red, shows that trend. You cannot really see it from the raw data, a trend that small, but you can see it from the bottom. So why does this matter? We basically made a piece of code in MATLAB, but it's what can be used as a tool. So SPG is a newer method. There's very little studying going on into it. But the hope is that much like PPG, it could eventually be used continuously for continuous monitoring. So using our code, you could look at brain activation-induced blood flow using SPG specifically. And this could have a number of effects later on. These are just a few examples that we came up with of how it could be used. Continuous monitoring for strokes, monitoring brain activity in patients with Alzheimer's. There's a number of different applications. So thank you to the Boston University College of Engineering, specifically Dr. Darren Roblier and Dr. Diane Joseph McCarthy for their help. Thank you to the Bio-Optical and Acoustic Spectroscopy Lab at Boston University and our advisor, Dr. David Boas. And thank you to Kilachand Honors College. Any questions?
Thank you so much. We have quite a bit of time for any questions, if anyone has any.
Thank you. That was a very impressive result you had at the end. I was wondering what areas you see for future work with this sort of method.
I mean, I kind of mentioned at the end a little bit, but anything where you want to look at brain activation. Obviously, a lot of labs, the lab I worked at is very interested in looking at how humans have different responses to stimuli, how their blood flow changes when you give them different stimulus, and then more practically in like a medical setting, like I said, it could be used in a wearable device like a watch or something like that, but for the brain, not like eventually something that could be wearable and continuously monitor for diseases like a stroke, in which you would see a significant decrease in brain activation blood flow.
Thank you so much. That was really interesting. Can you speak a little bit about what it was like to approach this project collaboratively, since you were working together with other people?
Yeah, it was really interesting. If anyone's ever coded in a group before, it's really not always easy to get 5 different people on one piece of code. We did a lot of splitting up. I mentioned there was a couple different ways we found the beginning of the beat, which was kind of the first big part of the project. I also mentioned the SVD, or singular value decomposition. That's not the only thing we tried there. We tried a lot of different types of that. So it was more at times of breaking it up and everyone trying their own thing, and then coming back together and regrouping and gathering our results and seeing what actually worked, what didn't work.
How does the sine wave map to the heartbeat? Like, people have different heart rates. So is this something that you calculate in the code that then is different for different people, or is it something you have to do beforehand?
So you wouldn't actually have to convolve the sine wave in, like, if you were actually using this. That was just to check our work. When we originally took the raw data, there should have been almost, like, the patient was at rest. There should have been little to no, like, stimulus, as much as we can control for anyways. So it should have been pretty flat. There should have been pretty much no brain activation changes. So we convolved the sine wave to check that it actually worked, that if we said there wasn't, like, a superficial brain activation change, would it actually track that brain activation change happened?
Congratulations on a great presentation. I was wondering if you could give an example of what would cause like brain activation-induced blood flow as someone like not as familiar with the topic.
So any type of stimulus, like it depends on which part of the brain you're looking at. If you're using I mean, it could be anything. You could be reading, you could be seeing something, whatever part of the brain you're looking at, really. If that's activated by whatever you're doing, then that'll change the blood flow. If you're looking more on like what we were looking at for applications, that would be more longer over time, less immediate.
Any other questions? Fantastic. Thank you so much for that incredible presentation.